Good morning. Today we will be discussing about sterilization with plasma. Sterilization using plasma, gas plasma, what I mean. And these days, in the era of pandemic, this word sterilization and sanitation has become part of life. So this is a very right topic to be discussed, and I'm thankful to the European Forum for giving me a chance to interact with you on this topic. So I will be starting with the basic concept. What do we mean by disinfection? Disinfection is cleaning an article of some or all of the pathogenic organisms that may cause an infection. But unfortunately, the ideal disinfectants do not exist. Whatever disinfectant are in the market, we will be seeing the labels 99% or 99%, 0.9% effective. But we have not seen any disinfectant which claims it to be 100%. And unfortunately, most of the disinfectants are by their nature potentially harmful and even toxic to humans and animals. The term which we often come across is cleaning. By cleaning, we mean that physical removal of foreign materials, example, dust, soil, organic material, such as blood, secretions, excretions, and microorganisms. And cleaning generally removes rather than kills the microorganism. By cleaning, we don't mean to that the microorganism will be destroyed, but it will be removed from the surface we are cleaning. It is accomplished with water, detergents, and mechanical action. And the term decontamination and sanitation is also used for cleaning only. The term that we are concerned today is sterilization. Sterilization, like in cleaning, I said that it's removal of the microorganism from the surface. But sterilization is a complete destruction of the microbial life, including bacteria, viruses, spores, and fungi. Before going for the strategies that we have for gas sterilization, we should know the factors that will influence our sterilization. The foremost factor are the type of the contaminating microorganism, whether it is bacteria, it is virus, spores, and fungi, and if possible, which kind of bacteria, which class of bacteria. Second is the degree of contamination. What is the count? How many bacteria are there? Whether it's a small contamination or large portion of the surface is contaminated. Third is amount of the protein material present. How many RNAs of the concerned microorganism are present. Presence of organic matter or another compound such as soaps may neutralize some of the disinfectants, and chemical nature of the disinfectant. The concentration and the quantity of disinfectant that we'll require and that we will be taking to disinfect the concerned surface, and the contact time and temperature. Of course, we will be generally working at the room temperature, and the contact time is crucial because must have heard that washing your hands with soap for 20 seconds is enough for this coronavirus. Somewhere you may get an information that for other microorganisms it may be 30 seconds, like there was a swine flu. For that, it was asked that you should wash your hands for 30 seconds. Next is the residual activity that effects on fabric and metal should be considered for the specific situations. The application temperature, pH, and interactions that will also affect the chemical reaction going on. We should also take into account that the chemical reaction should not be such that the surface that we want to sterilize gets destroyed. We are concerned with the sterilization of the surface, destroying the microorganism, not affecting the surface. Toxicity to the environment and relative safety to people that may be exposed. And the biggest thing is the cost. At what cost is this sterilization going to happen and how much effective it is? The cost of the sterilization process and the effectiveness are to be balanced. If we are going to have a radiation light source, then these are the factors that we just discussed. We can summarize those factors that we discussed right now by this pictorial diagram. The processes that we have for sterilization are broadly classified into physical, chemical, and physiochemical. Physical, that you all know, the direct sunlight, anything you keep in sunlight for a definite period of time will make it disinfected. It will sterilize it. Like in our Indian mythology, what our elder says, that fire purifies everything. By fire, we means that raising the temperature of a body purifies it. So it may be by sunlight, it may be by heat, direct heat, or raising the temperature by any means. Radiation, filtration, these are all the physical means. And we have chemical means like alcohols, which we are using these days. I think we are using it more than what is required and what was supposed to be. And the gaseous disinfectants. So right now we are concerned with the gaseous plasma. We will be limiting our discussion to gaseous plasma. And physiochemical are the combination of the physical and chemical. That is up to you to combine the two, any of the two, physical and chemical processes. And the combination should be such that it should not affect the surface. And what is plasma? How it came into existence? So, firstly, we should be knowing what we mean by the term plasma. So, in 19th century, Purkinje was a physiologist and he used the word plasma for the clear fluid that remains after the removal of the corpuscular material in the blood. The semi-transparent material that remains in the blood, that is called plasma. On the other hand, in 1922, Langmuir proposed that the electrons, ions, and neutrals that an ionized gas is composed of should be termed as plasma. Now, we are having a two kinds very difficult situation, because if you talk of plasma to a biologist or a doctor, they will certainly gather your views about the blood plasma, but you have to explain that we are not talking of the blood plasma, we are talking of the gaseous plasma. And the sterilizing property of the plasma was first introduced in late 1960s, and it was patented in 1968. And the biggest reason is that the sterilizing property came in 1960s because the ionization tools, the energy required for ionization, the ionization tools were into development up to that time. And after the development of laser in 1960s, this production of gas plasmas accelerated. Now, what is plasma and how I can obtain it? As we all know, in solids, we have a well-defined crystal structure. All the molecules and atoms are arranged in a definite lattice arrangement. And if we supply heat to solids, they become liquids. And if we supply more heat to liquid, that become gases. And more heat eventually ionize the gas into positive and negative ions. And neutral particles are also there because the gas is not fully ionized. And for the sterilization purpose, we will be focusing on the plasma having all the three components, that is, positive ions, electrons, and neutral atoms. And in the situation where the population of neutral atoms is quite large than the positive and negative ions, we term that situation as cold plasma. So, cold plasma is a plasma which is composed more of the neutral atoms and less of the ionized electrons and ions. So, the percentage ionization of the gas is very low in cold plasma. What are the factors that will affect the plasma sterilization? First is the choice of the gas. Which gas we are going to ionize? That will affect the sterilization process. The flow rate, at what rate the gas will flow, because the percentage ionization will also depend on the rate of the flow. The pressure at which the gas is there, that will affect the density of the plasma. The power also influences the electron density. Quantity of the material to be sterilized, because more the surface area, the more the plasma that you will require. The nature of the microorganism, the density and the surface layer formation, that is also required for the kind of plasma you require. And the packaging, the geometrical factors. The principle is that cold plasma is generated at atmospheric pressure by the gas through under the influence of an electric field. And the electrons that are arising from the ionization process are accelerated in this field and trigger impact of the ionization process. And free electrons colliding with the gas atoms transfer the energy, thus generating high reactive species that can interact with the contaminated surface. And the electron energy that is sufficient to dissociate this covalent bond in organic molecules. For single bond, the energy ranges from 1.5 to 6.2 electron volts. For double bonds, it is 4.4 to 7.4 electron volts. And for triple bonds, like a more higher energy, that is 8.5 to 11.2 electrons. How the microbial inactivation takes place with plasma is dependent on the ability of the atmospheric discharge cooled plasma to sterilize surfaces. And the combination of the electron and ion bombardment, thermal effect, free radical production, and local exposure. By local exposure, we mean the ultraviolet. All the above act in combination to disrupt the microbial cell membranes. The DNA structure, because in human body, microbial in an isolation is an RNA. It will combine with the DNA and it will be present in the DNA of the cell, human body. And this exposure to the gas plasma and the ultraviolet will lead to the changes in the microorganism structure and denature the proteins. The mechanism depends on three in three ways: the free radical interaction with the ionized species, the radiative effects of the violet or ultraviolet radiation, and the volatilization. The charged species in the plasma damage DNA formed in the vicinity of the domain, and the violet or ultraviolet radiation causes formation of thymine dimers in DNA, inhibiting the bacterial replication. They limit the population of the microorganism, and they damage the base and gradually the DNA strands breaks. So it dissolution of the DNA. By sterilization, we can sterilize the wastewater. We can sterilize the exhaust gases, vegetables, fruits, and these are the sterilizations that will be concerned with while dealing with the plasma medicine. We will talk about these all these in the when we will talk about plasma medicine in my next lecture. We have many processes so that plasma can be generated, but all of them are not applicable for cold plasma. The first source is the corona discharge. The plasma that is created by lighting a crown around the wire. That is why, due to this, it is called corona. Mechanism is very simple. This is a needle. You can take the metallic needle that we previously used to inject injections into human body and animals, the metallic one. A simple needle we can take. We can insert a tube into that needle, and the gas under high pressure flows through this tube. And on this metallic needle, we apply high voltage so that the gas which is flowing through the tube ionizes, and we get the plasma on the tip of the needle. It is high efficiency, low cost, and low operational cost. The advantages: noise, power loss, and insulation damage of the device. It is used for surface treatment for tissue culture and surface treatment of materials to change properties and sanitization of water. The next technique is the microwave induced plasma. Its frequency ranges from 300 megahertz to 10 gigahertz, and the commonly used wavelength is 12.24 centimeter, corresponding to a frequency of 2.45 gigahertz. It ranges from a few watt up to several hundreds of kilowatts, and the discharge pressure might range from less than 10 to 2 pascals up to several times of the atmospheric pressure, whereas many different discharge gases might be used. The advantages are the wide range of the operational conditions due to a vast range of the pressure and the frequency, and the clean as high chemical activity. It is used in waste treatment and surface treatment. And the third one is the gliding arc plasma. Here, the plasma glides on the two electrodes. That is why it is called gliding arc plasmas. And these two electrodes are diverging with respect to each other. They are diverging. They are divergent in shape, and the discharges start at the spot where the distance between the electrodes is the shortest and is spread by gliding along the electrodes in the flow direction until it disappears after a certain path to repeat the cycle. The advantages are the simplicity, low cost, environmentally cleaner, and applications are based for the plasma surface treatments. The next one is the dielectric barrier discharge. Here the plasma is created between the two dielectric surfaces, and generally it's an aluminum plate that we are taking, and that is connected to the dielectric. AC voltage electrodes and Teflon is generally used as a dielectric. We have two configurations. Either plasma is created between the dielectric surfaces like a sandwich, and in a single slab dielectric, we cut a groove and the plasma is generated in the chip form. You can see the plasma shapes in the two configurations. These advantages are high efficiency and cost effective, and the uses are food decontamination and surface treatment. The advantages are eco contamination of products, high cost effective, environmental and economically beneficial, pollution control applications, reliable and user friendly, mild surface decontamination technology for products such as cut vegetables and fresh meat, disinfect surfaces before packaging or induce as part of the packaging process. These are some of the things that I have recently noticed because in Hindi we say that avashakta hi avishkar ki janni. So these are some of the new products that have come in the market. This is an ultraviolet source. We can use it to sterilize the hospital, the offices. We can keep this source illuminated in the middle of the hall, and the light is and all the surfaces in the office or the hospital should be exposed to this light. So when we leave the office in the night and go to the office the next morning, it will is by that time every surfaces on the offices will be sterilized. The same based on the same principle, there is a sterilization of mobile phone. There's a gadget available in market right now, and it's a very low cost affair that you can have it. You can keep your mobile here, and while getting sterilized, it can also charge. There is a charging node also provided for, and the power comes through a USB cable, and that power is sufficient for charging the mobile phone as well to operate the UV tubes that are there. And a small device to sterilize the your glasses, wearing glasses, keychains, and all. This is also available in the market. The principle of these is very simple. There's a gas. The device which I showed in the last picture was a ultraviolet light, but there are also devices that is based on the gaseous plasma discharge that is being used for certain higher applications like on railway stations and airports to sterilize the luggage. There's a simple chamber, there's a gas flow, and this gas flow, there's a plasma created from this gas flow, and this plasma goes through the chamber, and ultimately there's an outlet. In the chamber, you can have your luggage placed for some time. Then that time depends on the size of the luggage, the plasma density that you are having, on the factors that we discussed previously. So definite time can be calculated, and that should luggage should be exposed for that time. The limitations are the cold plasma are the relatively early state of the technological development. And the important aspects of this technology are still immature. Optimization and scale up to a commercial treatment levels require more complete understanding of the chemical processes involved, and high investment in R&D is also a major cause. Variety and complexity of the necessary equipments, antimicrobial modes of action for various cold plasma systems vary depending upon the type of the cold plasma generated. So you have to very choosy in choosing the right gas and the plasma density. The future research is the optimization of technology, characterization of the entomicrobial compounds, the detailed examination of the sensory properties of the cold plasma, and more information on the economics of the process for large scale. And in the end, what the Donald Scott had said, because the world is started with the plasma, and whatever with the end, there will be plasma. So the plasma is everywhere, and it has to play an important role in